Welcome back to class. We are still in chapter 2. We're kind of right in the middle of chapter 2 now. We've talked about some different kind of reactions. We've talked about pH and water and solutions. And now we're going to start talking about some big molecules that our cells build. But before we talk about the big molecules, let's talk about the difference between inorganic and organic. We are not talking about what kind of carrots you're buying at the grocery store. We're not talking about garden principles. We're talking about the makeup of a molecule. And the way you tell if a molecule is organic or inorganic is you look at what's in it. If that molecule has carbon and hydrogen in it, let's change that to say and, put an and right there. If that molecule has carbon and hydrogen in it, it is organic. Our big kinds of organic compounds are carbohydrates, proteins, lipids, and nucleic acids. If that molecule does not have carbon and hydrogen in it, it is not organic. It is inorganic. These would be things like oxygen, water, inorganic acids, bases, salts, carbon dioxide. Without having both carbon and hydrogen, you are not going to build as big and complicated molecules. So inorganic compounds are usually going to be smaller and simple. So oxygen just has 2 little atoms in it. Water has 3 little atoms in it. Typically, a salt is only going to be 2 or 3 atoms in it. Whereas our carbohydrates, proteins, lipids, and nucleic acids can be like hundreds of thousands of atoms in them. So they're typically going to be called something like a biomolecule or a macromolecule because our organic compounds get really big. And on these big molecules, it's often a few specific regions of the molecule that are really doing a lot of the work. And those little regions are called functional groups. Functional groups are a little piece of a molecule that gives that give that molecule a lot of its chemical properties. So our organic molecules, again, they always have hydrogen and carbon. We're usually going to have oxygen in it. They're made of covalent bonds, and they have functional groups. Those functional groups determine a lot of its chemistry. So we see things like carboxylic acid. This is a carboxylic acid group, COOH. This can act as an acid. It can release this hydrogen here. And this is going to be important in fatty acids and amino acids. Every single amino acid ever is going to have this in it. And many fatty acids will have that in it. Amino groups can also act as an acid because they can release this hydrogen. It can also accept an acid, so it can act as a buffer, actually. It also can form bonds with other molecules. So this nitrogen is happy to have 3 bonds or 4. This amino group is going to be found in amino acids. It's what amino acids are named after. This hydroxyl group, or OH, is going to, this is what is the characteristic of alcohols, having this OH group. It can release this entire OH group as a base, and it can also be used in dehydration synthesis. So break off this OH while the other molecule donates a hydrogen, and so you're able to condense 2 molecules together. This is found in carbohydrates, fatty acids, and amino acids. Then your phosphate group, or PO4, a phosphate with 4 oxygens, but actually it should say O than PO3 because that oxygen is binding then to the phosphate and these other 3 oxygens. This is very important in energy. It can form high energy bonds and just hold a lot of energy in this bond where it's holding on. This is going to be important in phospholipids, so they're called phospholipids after a phosphate group, nucleic acids, and high energy compounds. If you've heard of ATP, ATP uses this to store its energy. When we build a large molecule, we typically build it as a repeating series of subunits. So those small repeating subunits are called monomers. If you are building a pearl necklace, the monomer would be each individual pearl. If you're building a block tower, the monomer would be each individual Lego block. Monomers join together by dehydration synthesis reactions, where you put 2 individual monomers together, and then you can add another one and another one until you get as long as you want. The first of the biomolecules we're talking about is going to be carbohydrates. Carbohydrates has carbon, hydrogen, and oxygen in it, and in a 1 to 2 to 1 ratio. So if you have 5 carbons in your carbohydrate, you'll have 10 hydrogens and 5 oxygens. If you have 7 carbons, you'll have 14 hydrogens and 7 oxygens. If you have 3 carbons, you have 6 hydrogens and 3 oxygens. That's an important principle. That's a way that you can identify something as a carbohydrate. So make sure that you're able to do this. If I tell you it has this many carbons, you could tell me how many hydrogens and oxygens in it. In a monosaccharide, you have a simple sugar. It'll be 3 to 7 carbon atoms. Not going to get bigger than that. Monosaccharides would be things like glucose and fructose. Your body uses them as an energy source. Plants can make it from photosynthesis. Things that need to eat, things like us, are going to obtain that energy from food. If you put 2 monosaccharides together, you have a disaccharide. So 2 simple sugars that a dehydration resynthesis reaction has put them together. So our monomer here is a monosaccharide, and now we're starting to build them together as a disaccharide. Disaccharides would be things like sucrose, lactose, and maltose. Sucrose being ordinary table sugar, lactose being milk sugar, maltose being malt sugar. These are all energy sources still, although your body is going to break them into monosaccharides before it uses them as an energy source. Then when you start putting a lot of them together, a lot of monosaccharides together, you build a polysaccharide. Polysaccharides can either act as a storage form of sugar. So glycogen is a way that we store sugar in our cells. Starch is how plant cells store sugar. Polysaccharides are also used to make structural chemicals. So the chitin that surrounds a fungal cell, the cellulose that surrounds a plant or an algal cell, those are also polysaccharides. They're structural versions of polysaccharides. And in bacteria, when we have peptidoglycan, that's a combination of sugars and proteins together, carbohydrates and proteins together. I think my cat is in the other room playing on the piano. I hope you can't hear that, and I'm sorry if you do. Okay, if you want to break down a disaccharide to a monosaccharide, or if you have a polysaccharide that you're breaking off monosaccharides, these are hydrolysis reactions. So carbohydrates, again, their principal function is to fuel your cell. They give you a source of energy. Although they're also used to build DNA. So each DNA monomer will have a little bit of sugar in it. They're used to build cell walls, we just talked about. And they're also important in building cell membranes. A lot of sugars will be attached to other molecules in the cell membrane. This is what glucose looks like if you were to just draw it out. Well, typically it forms a nice little ring. Here's the monomer, the monosaccharide. When we're looking at a polysaccharide, you can see lots of monomers put together. So here the monomer is a monosaccharide, and the entire biomolecule is a polysaccharide. Or you could draw it as a series of little dots, little circles instead of little hexagons. Okay, so now you can tell me what organic is. You can give me an example of a monosaccharide, a disaccharide, and a polysaccharide. You can tell me what they're doing. You can tell me how many carbons, hydrogens, or oxygens are in a monosaccharide. If I tell you either how many carbons, hydrogens, or oxygens are there, you can tell me the other two. Okay, I will see you back for our next lecture video where we're going to get into talking about our lipids. See you later.